Hello everyone, welcome to the St. Joseph's open web-based learning platform, that is how you learn. Also welcome to Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam. The Department of Biotechnology presents the SMOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. It's a free online modular 8-week course for life science background students. Will be offered at the UG level.
Welcome back to the course Plant and Animal Biotechnology, the third module, that is animal tissue culture techniques, involving the animal cell culture, culture of mammalian cells, cell viability, cytotoxicity, transfection methods, will be discussed very detailed by the Dr. A. Veerabramam, HOD, Department of Biotechnology, St. Joseph College for Women, Autonomous, Visakhapatnam. So these are the learning outcomes for the animal cell culture transcendences. Students can able to obtain information about animal cell culture, able to formulate the specific media constraints for a desired task. This is very, very important, as the student need to study the in-depth analysis of the each and every element that gives the a specific application and a specific a specific importance for the growth and development of the animal cells under desired conditions. Explore the significance of different tests in cell culture. So these are so once the monolayer or a single cell culture or a suspension culture is obtained, so whether these cells are live in condition or dead in condition, or live cell, how many live cells are there, how many. So both the cell viability and cell toxicity can evaluate the how many dead cells are there, how many live cells are there, can easily be evaluated through the variety of chemical analysis or a variety of reagents. And students can be an expert in different transfection methods, transferring the DNA molecules from one organism, or of course these methods are completely in vitro methods involving the calcium phosphate precipitation method, PEG mediated method, electrofusion, biologistics, gene gun method, shotgun method. So many methods are available for the transfection methods. Of course, involving the viral methods, also simian virus, SV40 virus involving method. So these are the contents for the present unit, that is animal cell culture media, culture of the mammalian cells, cell viability and cell cytotoxicity, transfection methods. First of all, discuss the animal cell culture media, cell culture of the mammalian cells. So for any type of the material, for example, any live conditioner or any plant, of course, as of now, the units deal with the plant material, plant cell culture, plant tissue culture, organ culture. So right now, this unit is specifically concentrating on the animal cells, animal cell culture, cell lines, especially how they can adapt to the cultural conditions. Of course, here animal cell culture involves most of the contamination problems, and specialized devices are required for the animal cell culture. CO2 incubator is required, and specially designed culture plates are required. Unlike plant tissue culture, here a specially designed media is required, and the media must be completely double time, it must be sterilized, then only it must be, and aseptically it must be transferred to the culture vials, and the culture vials are completely one in a time will be used once in a time, so that the culture vials are somehow expensive, but once the lines are established, then the animal cell culture field will be completely it's a boon for the studying the variety of problems associated with the human life. So animal cell culture involves the growing cells outside their natural environment under controlled conditions, often for research, drug development, or vaccine production. So these areas are hot topics as of now. Of course, every time these topics are very, very important. And transfection, the introduction of foreign DNA into the cells, is a key technique used in cell culture for various purposes. This explanation will cover the basics of animal cell culture, different types of cultures, transfection methods, and their applications. Animal cell culture basics, let us see what is the animal cell culture. It's a process of growing cells outside their natural environment under controlled conditions. Controlled conditions means a CO2 incubator. You need to provide the equimolar concentration of the oxygen and the carbon dioxide. So whatever carbon dioxide or a toxic environment, toxic gases or toxic environment must be replaced with the oxygen that need to be sterilized oxygen through a filtered atmosphere. Then only the animal cell culture can easily be established. So these are the culture media and metabolites, vitamins and minerals are required, and serum is required irrespective of other than the both organic and inorganic constituents. Here animal cell culture requires a serum completely and a pH indicator. Any slight difference in the pH of the solution animal cell culture media, that will be a problem for the animal cell culture establishment. The carbon source is required and buffering systems. A PBS buffer system is required. A phosphate buffer saline is required. And antibiotics are very, very important for the establishing or maintenance of the decontamination chances for the animal cell cultures. So this is the culture of the mammalian cells. Generally, cells are isolated from the organs and either mechanically agitated or enzymatic method. Cells are inoculated onto the specially designed culture vials and cultured under the aseptic conditions and desired conditions to get the confluent culture. That confluence means one layer of the cells, and then at regular intervals the cells will be subcultured, and based on the subculturing process the culture number is maintained, secondary culture or a primary culture. Once they are subcultured, the cell culture is known as the primary culture. So as many as times the primary cultures are subcultures, cell division occurs vigorously and continues after finite divisions. So as many as finite divisions are there, they must be called as the secondary cell lines. So these are called as cell lines. In such a way, any organ or having a tumor or normal organ, the same protocol will be applied to get the cell line. Okay, so in that way, from which the cells are originated, and the honor of this organ, the cell line is so named. Primary culture. Okay, this is a primary culture, this is a secondary culture, and these are the different culture vials. This is a rocks bottle, tea bottle, horizontal bottle, and sarcoma bottle. Okay, so this is the 2D two-dimensional cell culture. Once the cell lines are ready, those cell lines are septically washed with a buffer and again submitted or again inoculated or reared is the term used in the animal cell culture, and fresh media is every time must be added and incubated in a CO2 incubator to maintain or get the monolayer of the adherent cells. So in case of the animal cell culture, adherent cell lines and non-adherent cell lines. Adherent cell lines means those cell line is prepared, a specific cell line is prepared or a culture is prepared. Once it is obtained as a monolayer, these with the help of an anchorage, they can with the help of a substratum, not anchorage, substratum like a polypropylene sheet or a glass item, they can easily fix themselves onto the surface of the culture vial bottle. The culture vial bottle is, but every time it is fixed with the carbon dioxide oxygen exchangement, and two directions available for the cells to grow, the way of the cells and can grow. In 3D cell culture system, the marginal cells, non-adherent cells, most probably the medium is liquid media is used to rear or culture the lines and the organ. Here, the 3D level organ on a chip level based. Okay, so a chip level and a micro channels, the cells can also be each are linked with a centrally fixed shaft, and cells will be cultured. Let us see the types of culture in this chapter. Primary cultures isolated directly from tissues and have a limited lifespan. Those are known as primary cultures. Subcultured from primary cultures and can be propagated indefinitely, called as cell lines. Adherent culture, cells attached to this culture surface, culture vial bottle surface. Cells grow freely in the medium. Those are known as suspension cultures. Most of the blood associated cell culture systems are suspension culture. Most of the organ or tissues are culture under the adherent culture system. Cultural methods. So either of the monolayer cultures or a suspension culture. Cells grow in a single layer on the surface of a flask or a dish. So at this stage, the cells must not get any contamination. Then only the monolayer can be continued till the lines or desired patterns. Cells are maintained in a liquid medium and are not attached to any surface. Those are known as the suspension culture. Let us see what are the culture media required and culture methods. Animal cell culture involves cultivating cells outside their original environment and controlled conditions to study their physiology, genetics, and potential applications. This process depends on the specialized media and methods to support cell growth and viability. So let us see what are the cell culture media. Of course, in case of plant tissue culture medium, Murashigi, or even a single coconut water or tomato juice or basil salt solution can nourish any growth and development. But here, in animal cell culture, you need to design based on your target, based on your cell type, based on your cell line, based on the objective, project. Okay, so let us see what are the culture medias available for culturing the animal cells. Natural media, artificial media. Animal tissue extracts, blood, plasma, and other biological fluids were used as a media, that is known as a natural media. Provide a complex mixture of nutrients and growth factors. So this is a comprehensive media. Of course, sometimes may not give the exact confluence or a monolayer culture for all the types of the cells. But basically, some of the cells, for example, skin or blood, can easily be cultured. Today, most of the animal cell cultures are uses defined synthetic media. So based on the target, based on the organ, based on the place origin from which the cells are isolated, the media will be designed so that the target cells, lines or cultures will be obtained. These contain specific salts, amino acids, vitamins, carbohydrates like glucose as a carbon source, and often serum is supplement rich in growth factors. So in artificial media, there is a serum rich media or serum free media and attachment molecules. So types of artificial media, there are various types of artificial media. serum containing media, serum free media, chemically defined media, and protein free media. Okay, so each having a specific molecules in their difference in their establishment of the cultures for the different various purposes. So regardless of the specific formulation, animal cell culture media generally include energy source like glucose as a carbon source, amino acids as a nitrogen source, vitamins essential for various metabolic process, very important for a key process. Inorganic salts for maintaining osmotic balance and pH. Inorganic salts must be added as complex molecules. Serum, a common supplement providing growth factors, hormones, and attachment factor. Growth factors and hormones include the stimulate the cell proliferation and differentiation. Okay, so stimulation, cell proliferation. One cell must be need to double it in themselves and differentiate after line establishment after one one category of the lines created, they must be differentiated into or transformed into variety of lines. To prevent a microbial contamination, antibiotics are very commonly included in the animal cell culture media. And to maintain the stable pH, a variety of salts, lactic acid or dilute HCl can be used as a buffering system. Cultural method. Primary culture involves isolating cells directly from tissue and placing them in culture dish. Explant culture. Small tissue pieces are attached to a culture vessel and cells migrate out into the surface. Enzymatic dissociation. Cells are dispersed using enzymes like trypsin or collagenase and then culture. So in this case, trypsin is a is a good is an enzyme that detaches trypsinized, okay, or heparinized bottles are also culture vials are available to culture the animal cells. And cell lines. Established cell lines are derived from primary cultures and can be maintained in culture for extended periods. Adherent cultures. Cells grow attached to a surface. Example, plastic or a glass dish. Those are called as adherent cells. Suspension. Cells grow free floating on the culture medium. Most of the blood associated culture cell or in order to identify, study, or analyze, examine, suspension cultures are good choice. Cell culture techniques. Passages. Harvesting and transferring cells to new culture vessels to maintain a growing population, that is passage. Based on the passage numbers, the cell line viability or a potentiality will be determined. The more passage numbers, more culture, more rate. I mean, the cells are ready to give the give the potential to study or to examine any compound or any material. Maintaining cells under controlled conditions of temperature, pH, and gaseous environment, that is exchange of the oxygen to carbon dioxide. The next topic in the chapter is cell viability and cytotoxicity. Cell viability and cytotoxicity are key concepts in cell biology and biochemical research. In essence, viability indicates how many cells are still functioning, while cytotoxicity assesses the potential harm a substance can cause the cells. Okay, so based on the lethal dosage of any compound in animal tissue culture, the cells can be utilized. So that is a good choice and a good option in the animal cell tissue culture. Cell viability measures the number of healthy functional cells in a population. Okay, healthy population to find out the cell viability. It helps determine if a substance or a treatment is affecting cell survival or if cells are still able to perform their normal functions. So especially pharmaceutical drugs or new chemical entities can be evaluated or determine their concentration to prepare their drug for a disease. The cell viability functions test must be conducted. Cytotoxicity. that refers the toxic effect of substance on cells. At what rate a substance can control the normal physiological reaction, that is cytotoxicity. So its ability to cause cell damage, death, necrosis or apoptosis, programmed cell death, or to inhibit cell growth. So usually cells can every time, every hourly or every damage, the cells undergo normal cell division so that they can grow and develop and maintains the integrity of the body or a structure. So cytotoxicity is crucial for understanding the safety and potential harm of substances like drugs, chemicals, and medical devices. So cytotoxicity assay often involve measuring markers of cell death such as lactate, dehydrogenase, how much LDH is released because of because of the free radicals released by the cells, or caspase activation, or measuring the effect on cell membrane integrity once they are leaked because of this acidic environment. The plasma, the transient pores will be generated across the membrane so that the cell constituents will be leaked outside. Whatever the leaked constituents will be measured through the cytotoxicity. And transfection methods in this chapter defines it, the process of introducing foreign DNA or RNA into the cells. So there are two kinds of transfection methods: chemical transfection, physical transfection. Chemical transfection uses chemicals like calcium phosphate or liposomes to help DNA enter the cell. Physical transfection includes methods like electrophoresis, whereas electric pulse is given to create temporary or a transient force across the cell membrane for DNA entry. Biological transfection uses viruses like retroviruses to deliver DNA into the cells. Applications of the transfector. So usually transfection methods will be applied in genetic engineering. Two different sources of DNA molecules are united to form a mosaic or a hybrid, introducing new genes into the cells to study their function or produce desired proteins. Like retroviruses to deliver DNA into the cells. So that is of course the simian virus also naturally introduces the DNA molecules or RNA molecules into the cells. So in genetic engineering, that is genetic engineering, gene therapy, vaccine development, these are the applications of the transfection methods, introducing new genes into the cells to study their function or produce desired proteins. Gene therapy, treating genetic diseases by introducing corrected genes into cells. So this is a wonderful method in animal tissue culture technology. Animal tissue culture, gene therapy is proved to be our 99 affordable methodology to introduce a right gene to repair. Vaccine development, producing vaccines using cell culture and transfection techniques. So these are the categorization of the transfection methods: viral based, hybrid mode, non-viral based. Viral based, including stable transfection, retroviruses, lentiviruses can be used as a vector molecules to introduce the DNA molecule. And a transient transduction adenovirus can also be used. Adeno associated virus, herpes viruses can be used as they are impacts the exact of course the reliability on the viral particles may not be 100%, but maybe 40 to 50% reliability on the viral based transfection methods. Non-viral based methods, including the chemical and physical methods. In chemical methods, use of specific chemicals to aid the transfection methods. Either maybe micro or nanoparticles, polymeric based, or peptide or cations, calcium phosphate, dendrimers. So these are non-lipid based chemical methods. Lipid based, fusion of cationic lipid, liposome mediated transfection. Negatively charged genetic materials will be included in the lipid sac and directly injected into the tissue or organism. Physical or mechanical or non-chemical methods, electroporation, laser beam, gene injection, sonoporation, magnetofaction with metal coated nanoparticles or magnetic conjugated particles will be fired at a speed into the tissue. So these are the references for the present chapter. Thank you for your attention. Thank you very much for the present chapter, transgenesis.